All right, so this morning, or this afternoon, we have Dr. Nicolette Allred uh, presenting for us. She is a professor of biochemistry at Wake Forest School of Medicine, and her research focuses on metabolic disease, including type 2 diabetes, obesity, liver disease, nephropathy, and cardiovascular disease. She is involved in multiple large-scale collaborative projects, including the Genetics Underlying Diabetes in Hispanics, or the Guardian Consortium, where she holds an R01 to investigate the genomic basis of insulin resistance. She's also involved with Top Med, where she also holds an R01 to investigate the contribution of rare genetic variation to non-alcoholic liver disease. And she's involved with the GOLD Consortium, or the Genetic of Obesity Associated Liver Disease, where she's a member of the steering committee on the Consortium of Metabolomics studies. And so you can see she's very, very productive. She has 3 R01s and 172 peer-reviewed publications. So we're really excited to have you present for us today, Nicole, and we're looking forward to your talk.
Thank you, Rebecca. So I'm going to go ahead and start sharing my screen just to make sure everybody can see it. Can you see that okay?
Mm-hmm.
Excellent. So it's great. So thank you for that great introduction. And it's definitely a great opportunity for me to present the work from our group at Wake Forest with a bunch of friendly faces in the audience. So what I'm going to talk about today is metabolomics and metabolic disease and the utilization of contemporary approaches to identify biomarkers and understand pathophysiology in the setting of diabetes, although I won't focus on diabetes directly. And if my slides will advance, there we go. So to orient you to my talk today, I really want to begin with a discussion of the characteristics of insulin resistance, as well as a description of the measures that I will be focusing on today. So they're quantitative intermediate phenotypes, and I just want to make sure that we all have the same background. From there, I'll give you an overview of our prior work to examine the metabolomic architecture of insulin resistance, so kind of the basis for what I hope will be exciting new results, which will really set the stage for that modeling of insulin resistance using metabolomics with the expansion to additional cohorts. And then I'll include with some future directions for this particular project. So just to get us started, the most recent National Diabetes Statistic Report suggests that about 37.3 million Americans, or 11.3% of the population, has diabetes. And this equates to slightly more than 1 in 10 individuals. So this happy person right here in blue. Even more concerning than those statistics, another 96 million individuals, or 38% of the population, have prediabetes. And so this roughly equates to about 4 in 10 individuals. And this particular condition, if left untreated, often leads to type 2 diabetes within 5 years. And this is really where the bulk of my research focuses. So the southeastern United States really has the highest rates of both diagnosed and new cases of diabetes. And strikingly, these rates are higher in ethnic minorities who experience a disproportionate burden of disease. And to compound these issues further, the annual medical cost is estimated to be around $327 billion. So among diabetes comorbidities, heart disease is often the leading cause of death for both men and women, as well as people of most racial and ethnic groups in the US. And so the associated annual medical expense is estimated to be around $229 billion. And coronary heart disease, which is the most common form of heart disease, affects about 20.1 million adults, or about 7.2% of the population. So not only is it important to study subclinical indicators, but also diabetes and, leading to the incidence for heart disease as well. And so kind of all of these roll together for us to then form the basis for the insulin resistance atherosclerosis family study, or the IRIS family study. So in order to understand these underlying risk factors for diabetes, the IRIS family study was established in 1999 by the National Heart, Lung, and Blood Institute. And specifically, IRIS family study was designed to identify the genetic determinants of glucose homeostasis and visceral adiposity in minority populations. So ascertainment for this cohort was based on extreme family size. So we wanted really large multi-generational pedigrees and not necessarily on disease status. So for example, diabetes affects somewhere around 12% of our cohort, so equivalent to what we see in a population-based setting. So this particular cohort of just under 200,000, or 200,000, wouldn't that be nice? 2,000 individuals was recruited from 3 U.S. communities. Mexican Americans were recruited from San Luis Valley, Colorado, which represents a rural Mexican American population. San Antonio, Texas, which is a more urban Mexican American population. And then we recruited our African Americans from Los Angeles, California. And so this study will really form the basis for all of the work that I'm going to talk about today. So the data in Iris family study really exemplifies, I wonder why it fell off like that. Sorry about that. Really exemplifies big data. And so overall, our clinical measures include measures of glucose homeostasis as well as adiposity. And so I'm really going to focus on those from glucose homeostasis. So in order to assess insulin resistance in our study population, we've taken 2 complementary approaches. And so the first is really to assess those fasting indices of glucose homeostasis, which include things like fasting glucose, fasting insulin, and then using those 2 measures to create the homeostatic model assessment of insulin resistance. And all of these we really refer to as our basal models or our basal measures of insulin resistance. So these measures really mirror what is readily available in large epidemiologic studies. And so they give us really a foundation for comparison. Now, as a more sophisticated approach, we've also measured glucose homeostasis using the frequently sampled intravenous glucose tolerance test, or what we call the FSIGT. And this allows us to really capture dynamic measures of insulin resistance, which are due to some type of challenge. So we want to see how the body reacts. And so before I go any further, I just want to explain what those FSIGT measures are and kind of how they relate to the physiology. So type 2 diabetes is really a heterogeneous disorder that's characterized by both hyperglycemia as well as insulin resistance. And so both of those together really give us that overt disease phenotype. So beta cell dysfunction that you see over here on the left is believed to arise from islet beta cell degranulation and reduced insulin content. And this results in actually reduced levels of plasma insulin that you can see here. And in the large epidemiologic studies, this is what we would typically measure to assess beta cell function. So we would measure a fasting insulin value. In contrast, when we look at insulin resistant in the peripheral tissues, that really refers to that inability of the hormone insulin to elicit a response in the periphery. So this includes in the liver, the skeletal muscle, as well as in the adipose tissue. And so these tissues can't take up glucose from the bloodstream. And so in the setting of large epidemiologic studies, this would really be that increased glucose output. We would measure that as a fasting glucose measure. And so together, using these 2 measures, we could create that basal measure of insulin resistance, or that homeostatic model assessment of insulin resistance. Now, in contrast to that, our work really suggests that dynamic measures of glucose, those obtained from the FSIGT, may better capture the individual contributions of both beta cell dysfunction and insulin resistance. And so what we've done is we've measured what we call acute insulin response, or AIR, which is our first phase insulin release. And so this really characterizes that effect of beta cell dysfunction. Looking towards insulin resistance in the peripheral tissues, we've captured something called insulin sensitivity, or our SI value. And that's really looking at that response or that uptake of glucose from the bloodstream. And then when you put these 2 together, the product of these 2 measures is the disposition index, or DI, which captures the hyperbolic relationship to maintain glucose homeostasis. And in our studies, it's actually predictive of the development of overt diabetes. So this just gives you a background for the types of measurements that we're going to talk about in the studies that I present today. So with our clinical phenotyping, we've really recognized the power of dynamic quantitative traits to provide insights into disease processes. So as an example, I wanted to highlight this publication that came from Nature Genetics in 2009 that identified variants in the MTNR1B gene that were associated with fasting glucose levels. And this study was comprised of 10 genome-wide association studies involving up to 36,000 individuals, all of European descent. And so looking at this particular study, the strongest signal was actually observed at the MTNR1B locus for the variant RS10830963. And it was associated with an increase in fasting glucose levels with a very significant p-value, so times 10 to the negative 50th, so really stood out. This signal was subsequently attributed to association with type 2 diabetes in analysis up to 40,000 cases and 87,000 controls. But the p-value was a little less, so it was times 10 to the negative 13th, despite the increase in sample size. So this contribution is coming through fasting glucose levels, so an intermediate phenotype, and it's not as strong with type 2 diabetes. So in contrast, if you look at our studies in the Guardian Consortium, where we focused on dynamic measures of glucose homeostasis, particularly looking at acute insulin response, which is similarly measuring those beta cell dysfunction, looking at fasting glucose levels similarly, we actually see a significant association with AIR. So in 4 genome-wide association scans involving up to about 2,500 individuals of Mexican descent, we see the same association signal associated with a decrease in beta cell function, so 9.5 times 10 to the negative 12th, despite more nominal associations with fasting glucose in our population. And so this really tells us that our dynamic measures are capturing some additional features of glucose homeostasis. And our significance, which is genome-wide in this case, is really interesting because we're currently at about 1/14 the size of that seminal report. And so this really tells us the value of those quantitative intermediate phenotypes, especially those dynamic measures. So there's really added value in exploring dynamic measures of glucose homeostasis. So beyond direct metabolic or clinical measures, as a molecular genetics, I'm also interested in genetic variation. And so our genetic assessment of individuals in the IRIS family study ranges from candidate gene studies, which may include about 4,000 variants overall, all the way up to whole genome sequencing. So we've sequenced approximately half of our cohort, and we have about 25 million variants in those individuals. And so that really makes for a great comprehensive genetic data set as well. And then finally, we've also been exploring small molecule metabolite profiles, or what we term as metabolomics. So metabolomics is really the systematic study of the unique chemical fingerprint of the cell, that cellular processes that are occurring within the cell leave behind. And so to date, we've amassed a number of complementary panels. However, today I'm really going to focus on the HD4 discovery panel, which includes about 1,274 metabolites. And all this data was generated by Metabolon. So as metabolomics is a new approach in the study of common complex disease, I wanted to put this particular omics technology into perspective. And so looking at the slide, we really kind of had that central dogma. So DNA to RNA, RNA to proteins, and then the resulting interactions produce those metabolites. So what you're seeing really with the DNA level is the ultimate potential of the cell. So what is actually possible based on that genetic code? And then moving along with the RNA, kind of the next level of regulation, we're looking at that current direction of the cell. So what actually is happening versus at the protein level, using proteomics, we can assess the functional capability of the cell. So see what actually makes things happen. And then at the metabolite level, this is going to be the limiting currency of the cell. It really tells us what is happening right now. So based on sheer numbers, it may actually be simpler to look at the metabolome than actually the proteome, or even if you take it all the way back to the genome, just because of the sheer amount of the data that you're looking at. So with the metabolites, we're looking at about 1,274 metabolites in plasma, versus if you do whole genome sequencing, we're looking at about 25 million variants with DNA. And so this may actually provide more data. And because metabolomics is telling us kind of what is actually happening in the cell, it may give us more information that's actually proximal to disease processes and closer to those quantitative intermediate phenotypes. So the metabolomics data that we're looking at are from Metabolon, from the Discovery HD4 Global Metabolomics platform. And so what you're seeing in this figure are just the super pathways that are captured. So we have amino acid, cofactor and vitamin, nucleotide, carbohydrate, energy, lipid, and then we also have some novel metabolites, and these can include some partially characterized species as well. So these metabolites are dynamic in nature. So all of our study participants in Iris family study, we obtained their plasma sample after an overnight fast just to give them all kind of that standardized background. And we expect that these metabolites will really be more proximal to the disease phenotype, as I suggested on the previous slide. So the first thing that I want to talk about in terms of actual kind of results and research we've done is just kind of our work that really began with a publication in 2018, where we used metabolomics to identify distinct signatures of glucose homeostasis using our Iris family study population. And so the goal of this study was really to provide a comprehensive survey of the metabolomic signature of dynamic measures of glucose homeostasis using metabolomic profiling in our Mexican American participants. And what we did in that publication is we actually compared it with those basal measures just to see what are the differences really if we looked from a metabolomic centric perspective. So the premise of this study was that the metabolome is a biochemical fingerprint that when coupled with clinical phenotypes can provide insight into pathophysiologic processes. And so our guiding hypothesis for this work was that the addition of untargeted metabolomic data to sophisticated measures of glucose homeostasis will synergistically accelerate our understanding of the etiology of insulin resistance as well as its contribution to cardiometabolic disease in a rapidly growing US minority population. So all the results that I'm going to focus on here are driven from the Iris family study, but really focusing on our Mexican American cohorts from San Luis Valley, Colorado, and from San Antonio, Texas. So very briefly, our statistical analysis approach included outcomes, which were our basal and dynamic measures of insulin resistance. So for our basal measures, we included our homeostatic model assessment of both insulin resistance and beta cell function. And then for the dynamic measures, we included AIR, SI, and then DI. But I'm really going to focus today on those comparisons with insulin resistance. The way we set up our analysis, our predictors were our metabolites. And for our metabolite significance, we actually used a strict Bonferroni correction based on the number of metabolites examined. So this represents really correction for all available metabolites despite existing correlation between some of those. And then of course, in all of our analysis, we accounted for a minimum set of covariates. So these included things like age, sex, BMI, and then we also included recruitment center because we wanted that to reflect kind of that rural versus urban environment. And so to do this analysis, we used a variance component model as implemented in SOLAR. We used that to test for association between metabolites and glucose homeostasis phenotypes. And that really allowed us to count for that familial correlation using a random effects model. So in Iris family study, we have those multi-generational pedigrees. And so we needed to be able to account for that so that we didn't have any spurious findings. So this next series of slides that I'm going to present are really the results of analysis. So before I start discussing them, I just kind of want to orient you to this slide. So this is very similar to what you've seen before with a Manhattan plot, except now instead of genome-wide coordinates, we're actually using super pathways along the x-axis. So you can see those pathways of metabolism that I talked about before, amino acid, carbohydrate, vitamins, energy, lipids being our largest class, nucleotides, peptides. We have some xenobiotics as well as the unknowns, which are kind of those partially characterized species. I will say that our unknowns have been really interesting for us. So these don't represent kind of sporadic signals, but rather things that are consistently seen in the spectra. So these are likely real. It's just they don't have, a control sample to run against them, to characterize them. So along the x-axis, like I said, these are going to be our super pathways, and then we're going to have our significance on the y-axis. And this is going to be the negative log base 10 of the p-value, so that things that are at the top are the most significant metabolites observed. And so what we're looking at here is really insulin sensitivity, that association of each individual metabolite represented by these data points. And so the first observation that we were really proud to see was that we saw a lot of associations that kind of recapitulated what we expected from the literature. And so we know that metabolites associated with branched chain amino acid metabolites are really associated with type 2 diabetes. And so it was published in Nature Medicine back in 2011 that fasting concentrations of these amino acids are elevated up to 12 years prior to the onset of diabetes. And the risk for future diabetes was elevated at least fourfold in those with high amino acid concentrations. And so branched chain amino acids being leucine, isoleucine, and valine, and their intermediates. So that was really interesting, but you'll notice we saw a lot more associations as well. And overall, there were about 99 significant metabolites that reached that strict Bonferroni correction. So a more interesting or a more striking observation that we saw was really that signature of long chain fatty acids, which was not previously documented. And so this is stearate, margarate, and palmitate that you're seeing were highly associated. And these really suggest, so you can see that it's associated, just this text taken out from our paper, the insulin sensitivity was associated with 99 significant metabolites, and those explain approximately 28% of the variance beyond that explained by BMI. And the most significant were really those branched chain amino acids, which was that novel signature that we, beyond branched chain amino acids, was long chain fatty acids, which was really that novel signature that we observed. So in short, these findings really suggest that insulin resistance is metabolically complex. I mean, there's 99 significant metabolites associated. And in comparison, if you look at something like acute insulin response, there's really only a handful associated, with the most significant being glucose, of course. So through the use of metabolomics, we now have some insight into potentially why there's really a deficit of genes associated with insulin resistance. It could be the metabolic complexity. So apart from the increasing presence in the general population of insulin resistance without overt disease, insulin resistance itself really appears to be a very heterogeneous phenotype. And so one of the other things we've looked at towards kind of disentangling the contribution of adiposity, which has really been thought to drive insulin resistance, is we actually compared models with and without BMI included as a covariate. So as seen here, this model is without that seen with BMI, and you can see we still see some association with our branched chain amino acids, not the same significance that we had with our long chain fatty acids. But when we actually go ahead and adjust for BMI in this, we actually see that our branched chain amino acids actually reduce in their effect, which is consistent with them coming through a more obesogenic pathway. And then our long chain fatty acids actually come up as more significantly associated. And so this to us suggests that BMI is a contributor, but maybe not a main driver of insulin resistance. So this is just seen here in tabular form. We looked at the proportion of variance explained by demographics and or metabolites for those measures of glucose homeostasis. This was looking particularly at insulin resistance, where we looked at 922 samples. And in a minimally adjusted model, so only accounting for age, sex, and center, we account for about 6% of the R squared. And this actually increases to 35% with the inclusion of BMI, and then to 63%, so almost double, and with inclusion of those associated metabolites. So those metabolites really explain a significant proportion of the variability in insulin sensitivity. So it does suggest that these demographic characteristics, so minimally age, sex, and center, as well as BMI, when we couple them with metabolomics data, they could actually be used to estimate insulin sensitivity in additional populations, using as our model the results we see in Iris family study. And finally, when we compare basal measures with those dynamic measures of glucose homeostasis. We're looking here at an opposed plot. So very similar to what we saw before, and on the positive y-axis, we have our insulin sensitivity, and then on our negative y-axis, we have HOMA-IR, so two complementary measures of insulin resistance. And what you'll see here is there are striking differences observed between SI versus HOMA-IR in terms of, for example, branched chain amino acids, our long chain fatty acids that we talked about before, as well as some phospholipids here too. And so it's really striking that these overlap, but not exclusively, suggesting that insulin sensitivity may provide additional insights into the biological diversity of glucose homeostasis beyond what we see at the fasting level or at that basal state. So coming up, so what exactly did we take from this study moving forward? So these are some of our observations. So we really think that insulin resistance is metabolically complex, and that the complexity actually increases for measures of insulin resistance compared to those for insulin secretion. So there were very few metabolites that were associated with insulin secretion as compared to either SI or HOMA-IR. Secondly, BMI is a contributor, but not the only driver of insulin resistance. So insulin resistance is not exclusively attributed to obesity. And finally, when considered in the context of large epidemiologic studies, dynamic measures of glucose homeostasis may actually provide additional insight into the pathophysiology and identify contributing pathways. So taken together, our results really provide insight into those biological mechanisms of insulin resistance, and they could represent early pathophysiologic changes to enhance both risk prediction as well as improve diagnosis and treatment of type 2 diabetes. So where do we really go from here? So we've taken a number of different paths. So this really represents our fork in the road. So the first thing that we've done is, building upon these findings, we've used these statistical approaches to integrate metabolomics, metabolic or clinical phenotyping, as well as genome variation to characterize partially annotated metabolites and assess their role in cardiometabolic disease. So this graphic abstract really shows kind of our published work with metabolomic lactone sulfate, in which we identified that genetic predisposition through DNA, RNA, protein, all the way to our metabolite levels. And we found that when this particular metabolite, which was one of those that are partially characterized or termed unknown, was actually increased, we actually saw increases in adiposity and negative associations with cardiometabolic disease, including insulin resistance, dyslipidemia, as well as hypertension. And so that's one additional kind of study that's kind of flanked off from this that we published earlier this year. We also currently have a grant to explore machine learning approaches for metabolite estimation to examine contributors to glucose homeostasis and adiposity in the genetics underlying diabetes in Hispanics, or the Guardian Consortium, which includes IRIS family study. So I recently presented our preliminary results as an oral abstract at the American Diabetes Association. And what you see here is kind of our overall study design. This is in collaboration with Hakeem and her team at the University of Chicago that we reimagined her PredictScan framework. So this was used in the context of transcriptomics, but for this application, we're actually inputting our genetic variation data from our existing genome-wide association study, and then adding to that our metabolite levels. And so together, using that PredictScan framework, we're actually able to create models that actually use genetic data to predict that genetically regulated component of metabolomics, and then examine those predicted metabolites for correlation with our clinically measured traits. And so this is really fruitful in cohorts that lack empiric metabolite data, which is the majority of the cohorts that are available in the Guardian Consortium. And so this is a proof of principle concept where we're looking to do this in, right now, just Mexican Americans, but ultimately extend that to other populations as well. And then finally, what I really want to focus on for the last part of our talk is looking at machine learning approaches to estimate the contribution of insulin resistance to common complex disease. And I'll just say that this is a resubmission of an R01 that was initially reviewed by KNOD. It had a good score but wasn't fundable, so we're trying to address some of those reviewer comments. And so this is really the work that we have to look at insulin resistance. So actually estimating that measure using our metabolomics data. So this actually doesn't take advantage of the genetic data we have available. So insulin resistance is really recognized as a major risk factor in the development and progression of type 2 diabetes. Not only is it the most powerful predictor of future development of the disease, but it is also a therapeutic target. So once hyperglycemia is present. In addition, insulin resistance has been implicated as a significant contributor to multiple common diseases, such as cancer, cardiovascular disease, depressive syndromes, and Alzheimer's disease. However, with very few exceptions, the relationship of insulin resistance with that of any of these disease risk factors is really poorly understood. And a major barrier to effectively measuring that contribution of insulin resistance is the difficulty in acquiring high quality measures when the most desirable data actually requires invasive and very expensive examination in a clinical research unit setting. So although the cause of insulin resistance is still unknown, it has a close correlation with obesity. So there is increasing evidence that fat distribution, especially in the abdominal area, is correlated with the most severe states of insulin resistance. Therefore, it's important to take into account kind of that total body fat, which also may play a role. But as I showed you previously, it's not the only driving factor of insulin resistance. So we also want to consider that metabolomic architecture as well. And one way we can do that is actually measuring or estimating these quantitative measures of glucose homeostasis. So the premise for this study was that insulin resistance is constantly invoked as a contributor to disease, but with the exception of a narrow segment of diabetes-related studies, it's infrequently measured and rarely measured well. So we typically don't have these sophisticated measures of glucose homeostasis as we exemplified in IRIS family studies such as the FSIGT. So our hypothesis was that comprehensive metabolomic phenotyping can be used to build statistical models which accurately estimate insulin resistance. So what we're doing is we're taking those metabolites that we measured using the 1274 and trying to figure out the combination that yields the best estimation for our quantitative measures of glucose homeostasis. And we're going to explore both dynamic measures, so that would be insulin sensitivity, and our basal measure, which was HOMA-IR. Insulin resistance from multiple protocols, so either fasting or the FSIGT, can be estimated in large population-based cohorts, so now extending that outside of IRIS family study, that lack empiric measures of insulin resistance. And this allows us to directly measure its contribution to common complex disease. So we could actually look at it in the setting of Alzheimer's disease, cancer, cardiovascular disease by estimating insulin resistance and then assessing its direct association. So the study design for this work is a little different and includes not only our IRIS family study Mexican American cohort, but the first iteration, which is limited to our Mexican American cohort, includes San Luis Valley, Colorado, which we set up as our training set, and San Antonio, Texas, which is going to be our testing set. Then in our second iteration, we actually combined these two into a single training set in our Mexican American cohort. And then we had our testing set now including our African Americans from Los Angeles, California. So because this approach is a little different, I just wanted to show you how these participants compare demographically. And so in the combined sample, the mean age was about 40 years, plus or minus 13 years. They were 59% female overall, and the participants were generally overweight. So you can see the BMI was around 27 to 29. Looking at the individual centers, what we saw was our San Antonio Mexican American population And our Los Angeles African American population had slightly higher BMIs than what was seen in our San Luis Valley, Colorado population. San Luis Valley also had higher dynamic insulin resistance measures as compared to lower observed in both San Antonio, Texas, and our African Americans from Los Angeles, California. And you expect really you're kind of seeing this inverse relationship, higher versus lower. And that's because insulin sensitivity is measured by SI versus insulin resistance, which is measured by IR. So you kind of get that reciprocal effect. So that was our overall study design. So I'm just going to take that first iteration. So the Iris family study Mexican-American sample consisted of identically ascertained and examined participant from the two recruitment sites. And so those were San Luis Valley, Colorado, and San Antonio, Texas. And these samples were approximately equivalent in size. And so we just arbitrarily decided that San Luis Valley would be our training set and San Antonio, Texas would be our testing set. And that allowed us to keep our pedigree structures intact. Alternatively, we could have cherry picked pedigrees from each and combined them. That's definitely an alternative approach we could use. And so we treated San Luis Valley as the training set. Metabolite data were then fitted in a regularized regression model where empiric SI was really that outcome measure. And then we wanted to test how close our estimation came to that. So this figure really simplifies our overall study design. So first, our metabolomics data from San Antonio was transformed using 3 different approaches to optimize estimation. So we used standardization to rescale the metabolite data to have a mean of 0 and a standard deviation of 1. We used the inverse normal transformation. And with this, we're looking to get a normal distribution, a rank-based normal distribution of our metabolites. And then with the last, the Box-Cox method, that transformation was used to identify the best transformation for each metabolite individually. So we could have many different transformations across our data set. From there, we used a 10-fold cross-validation to select the Beck's mixing parameter in the 2 regularized regression models. And so the least absolute shrinkage operator, or the lasso, and the elastic net were used. So we randomly selected 90% of the data for developing our model to select the optimal set of metabolites for inclusion. And then the remaining 10% were set aside and used as a testing set. So this procedure is then repeated 10 times for that 10-fold cross-validation. And then from there, the selected metabolites from the regularized regression model, as well as our covariates, so age, sex, and BMI, were fit in that mixed effects model with kinship covariant structure and our random intercept to account for family structure, because this is, large pedigrees. And that allowed us to estimate our insulin sensitivity value and then compare that with the empiric or directly measured values in the San Luis Valley and the San Antonio cohort. So the correlation between the estimated and the empiric SI in either San Antonio or San Luis Valley were calculated using a Spearman's rank correlation coefficient. So I've described the kind of study design here, focusing on just our application in our Mexican American populations, but the same approach was used in applying our models that we build in Mexican Americans and looking for how they perform in our African Americans. So these are the results from our first analysis, where we trained the model in San Luis Valley and then applied the model to San Antonio. So you're looking at our training set here, which is San Luis Valley, and then our testing set, which was San Antonio. And then in each of these values here, we're actually measuring that correlation coefficient between the estimated and empiric SI. So the 4 models that we evaluated included Model 0, which was just age and sex, Model 1, which included, in addition, BMI, Model 2, which included age, sex, and associated metabolites, and then Model 3, age, sex, BMI, and associated metabolites. So Model 3 really was our overall model, or where we really derived our primary inference. So for simplicity, I'm focusing on results here exclusively from the lasso and the data that I'm going to present on the next couple of slides, although similar results were obtained from elastic net as well. So in the San Luis Valley cohort, so in our training set, in looking at our metabolites, we observed 49 metabolites that were associated when we did an inverse normal transformation, 37 associated from a standardized approach, and 48 from a Box-Cox transformation. And those were associated with our empiric measures of SI using that lasso approach. So looking at the correlations, these transformations that I show here really had minimal impact on differences in performance across the model. So you can see they're pretty consistent across whichever model you look at. So what we're looking at here in this final model in model 3, that included age, sex, BMI, and our metabolites in the prediction model. And it really had an improved estimation and resulted in a correlation of around 0.77 to 0.78 between empiric and estimated SI. And so that was really good for a first pass of the data. Looking here a little further, we also have results where we trained our models looking in our Mexican Americans and then applied them to our African Americans. So the data that I'm showing here is still the same. And what you can see, if I just pull your eyes immediately over here to model 3, you're looking at that Spearman's rank correlation between empiric and estimated SI now in our African American cohort. And what happened was in our Mexican American, we actually increased when we combined San Antonio, Texas and San Luis Valley, Colorado, we actually increased the number of metabolites that were associated. So then those metabolites were used for prediction in our African Americans. And they indeed gave us Spearman correlations that were on average about 0.72 to 0.74, depending on the transformation of the metabolites used. So what we're really taking from this data is that cross-ethnic validation worked relatively well with correlation between empiric and estimated SI, which ranged up from about 0.72 to 0.74. And these models were very similar to what we saw with elastic net. So right now I've been focusing really on insulin sensitivity as our dynamic measure of glucose homeostasis. So we next evaluated in the context of insulin sensitivity, the performance of estimated SI compared to empiric, looking at correlations with other clinical phenotypes in our data set. And so for this, we chose body mass index or BMI, low density lipoprotein or LDL, and our triglycerides. And if we look first at empiric SI, so this is directly measured in our cohort, we see that SI was negatively associated with BMI, with LDL in our African Americans only, and then with triglycerides in both our San Antonio population and our African American cohort. So you can see only LDL was non-significant, and that's why I've highlighted that p-value there. But the others had a p-value less than 1 × 10⁻³ . Similarly, estimated SI displayed the same patterns and effect sizes. So if we look at estimated SI using the lasso method, you can see that the values for estimated were similar to empiric, as was the significance level. So still looking at SI, we didn't have association with or a significant correlation with LDL, but that was really expected from our empiric data. So this suggested us that the estimated SI not only is being estimated well based on the correlations, but we're also seeing similar biological relationships with other clinical phenotypes available in our sample. So those were the results for SI. These show the results for that basal measure, HOMA-IR, which is complementary, although it is kind of that inverse relationship. And so you can see here, looking again, just drawing your attention to model 3, when we use San Luis Valley to predict into San Antonio, we see that the Spearman correlation ranges from 0.71 to 0.72. And so we're still getting really good estimation using a different set of metabolites here, metabolites associated with HOMA-IR. And then when we try this to go cross-ethnic validation from our Mexican Americans to our African Americans, you can see that high but slightly less. It's around a correlation of 0.66 to 0.68. So our correlation works well both within and across ancestries. It's probably even better than what we expected with our first pass analysis. Similarly, comparing the performance of estimated HOMA-IR now to empirically measured HOMA-IR and looking at that correlation with additional clinical phenotypes, what you can see now is looking at empiric HOMA-IR, we see strong association with BMI across both cohorts, as well as with triglycerides, and then LDL was associated only in our African American cohorts. And I'll just note that these correlations are positive now due to that inverse relationship between HOMA-IR and insulin sensitivity. Looking at the estimated levels, again, consistency in the models, you're seeing really the same effects for the most part in terms of that correlation between estimated HOMA-IR and directly measured BMI, LDL, and triglycerides. And so this really displayed the same patterns and effect sizes across cohorts using empiric and estimated data. So some conclusions from this work, which is really still evolving, is that metabolomics in combination with conventional clinical measures, estimated SI and HOMA-SI, with a high correlation between those empiric and estimated measures. And so although there is still room for improvement, which could include additional or different measures of adiposity, so you can think about waist circumference or waist to hip ratio, we could consider higher order interactions among our metabolites, or we could consider additional machine learning models. Towards utility, insulin sensitivity, which is a dynamic measure of insulin resistance, and HOMA-IR, which is our basal measure, were both estimated from the same metabolomic data, which was derived from a single fasting plasma sample. So this really represents efficiency of approach. So we could actually estimate these from a single sample as opposed to, for example, in the FSIG, taking multiple samples during a very long clinical visit. And our preliminary results suggest the estimation models worked reasonably well across race and ethnic groups, although a multi-ethnic model will likely be necessary. So overall, applying this method to estimate multiple methods for measuring insulin resistance from a single clinical visit may actually be valuable and provide insight into the relationship of different insulin resistance measures and their relative value for predicting risk of overt disease. So our next step will definitely be to apply these to various disease states, like I mentioned at the beginning, where insulin resistance has been implicated as a potential contributor, but not necessarily quantified. So how do we plan to move forward with this? So of course, our first step is resubmission of that R01, and we include expanding our measures of glucose homeostasis. So beyond basal and FSIGT, we also want to include the Matsuda index from the oral glucose tolerance test and MNS from the euglycemic clamp. It will also be informative to expand these studies to additional race and ethnic groups, particularly European Americans, if we want to create that multi-ethnic model. And so we already have collaborations in place with cohorts to do that. And finally, our ultimate goal will be the application to multiple disease states where we have already established collaborations to look at cardiovascular disease. So this is in both MESA and ERIC. Alzheimer's disease with Corinne Engelman, as well as Suzanne Kraft here at Wake Forest, and then with pancreatic cancer with Rachel Stoltenberg at NIH. So in closing, this research would not be possible without a collaborative team of investigators and of course our funding sources. And we're always looking to expand our collaborations. And with that, I'd be happy to answer any questions. I can't hear you, Rebecca.
Thank you so much for a great presentation. Yeah, I really enjoyed it. So we have one question in the chat from you from Shahjahan Ali. He said, excellent presentation. What do you recommend to reduce the burden of insulin resistance across the US?
So I think a lot of things that we can recommend without pharmacologic intervention would be things just like dietary modification, and that's coming from that contribution of obesity to insulin resistance. But I think if we were able to estimate insulin resistance, and if you look at some of our earlier work, we actually see a really high, using some of our IRIS Classic data, a high prevalence of insulin resistance in the general population. It's just not exceeded the level that we're able to compensate for with insulin release from the pancreas. So there's definitely that underlying insulin resistance, but I think metabolomics would be helpful to point at those people that are really in risk of that dysregulation, which could, ultimately convert to type 2 diabetes. So, I mean, short answer, I think the diet would be the quickest, diet and exercise to get at that obesity component, but then thinking about metabolomics is going to be more difficult because there's multiple metabolites from different pathways that are contributing. So you would have to augment those individually.
Makes sense. Thank you. So I have a question. So now when you go to the doctor, they can, when they do tests, they can give you like your genetic risk of heart disease, your genetic risk for certain lipid disorders. How far are you away? Think we are away from them doing things like this with proteomics?
With metabolomics?
Metabolomics, sorry.
I think we're still a ways away. So we're not doing metabolomics for this particular application because it's more discovery based. This is currently not in a CLIA environment with return of results. And I think it really requires more interpretation. So we know what, the biological characteristics of the metabolite, but beyond that, I don't think we know a lot about them. So the metabolomic lactone sulfate story that I mentioned briefly, that's a really good example where we started with a metabolite that was partially characterized and implicated in cardiovascular disease. And our next steps are to understand how you would augment that to promote cardiovascular health. The problem really is that we have to do that individually for each of those metabolites, which takes time. So I think until we have some more generalized suggestions, it may be more difficult to prescribe those as kind of risk assessments.
Okay, yeah. So, well, it looks like you've made really good use of all of these epi studies that were data was collected years ago. So, it's important, I guess, to collect those samples and store them. So,
Right. And so, I mean, so there's a couple of different approaches that we've taken. So if they have samples available, we can measure metabolites and estimate insulin resistance. And then for other studies, which actually don't have metabolites available, but have genetic data, we can actually use that to estimate the metabolites. So I think we're kind of coming at this from both sides to inform the study.
thank you. Are there any other questions for Dr. Allred? Well, I really appreciated it. Thank you so much. Thank you.
Thanks, Rebecca. I hope everybody has a great afternoon.
You too. Thanks. Bye-bye.
Nice job, Nicole.
Thank you.